Hello students, how are you? I hope all of you are fine. Okay, this is the next lecture of enzymes and we will be studying about cofactors. We will be studying about the types of cofactors that are present inside the body. What we are going to do first of all is let me go to the whiteboard and then I will tell you about the cofactors. Okay, let us just see this one first. Cofactors are special types of chemicals, special types of radicals. They are of various nature, which generally the function of the cofactor actually is to attach with the enzyme and activate the enzyme and give the enzyme strength to work. In most cases, sometimes the enzyme is unable to carry out the reaction. In that case, we say that the enzyme is in an inactive state. The enzyme is inactive and it needs some help. And the helper that is going to come to help the enzyme is the cofactor. This cofactor is actually a non-protein component. The cofactor is a non-protein component, and this cofactor, it attaches with the enzyme. It helps the enzyme activate the reaction, and then it most of the time it leaves the enzyme. I am saying most of the time. Why I am saying most of the time is because in every case, not in every case, does the cofactor leave the enzyme. We'll go forward and I'll tell you the examples of this. Right? Now, an inactive enzyme is known as the apoenzyme. It is known as the apoenzyme. And when this apoenzyme, it attaches with the cofactor, then this apoenzyme, it is converted into the holoenzyme. The holoenzyme is the active state of the enzyme. This holoenzyme is actually the active state. Apoenzyme is the inactive state of the enzyme. Plus the cofactor, it is converted into the activated state, and that is actually the holoenzyme. It is actually the holoenzyme. Now we have some enzymes that are generally, they are secreted in inactive form. For example, pepsinogen. Pepsinogen is secreted in inactive form. Pepsinogen is the enzyme that is mainly responsible for the starting of the protein digestion in the gastric cavity, in the stomach. It carries on protein digestion in the stomach, but pepsinogen, it is secreted in inactive form. Why is this pepsinogen is secreted in inactive form? Because it has an extra amino, it has an extra polypeptide extension. It has an extra polypeptide extension on the active site. This polypeptide extension has to be removed to activate it. Now, why is this polypeptide extension present over there? It is present there because in the absence of protein, if this enzyme is secreted in active state, there are chances that it can damage the tissues of the stomach. So this is a protection from God that this polypeptide is present there in the active site and it renders the pepsinogen inactive. This pepsinogen is then, if protein is present inside the food, then gastric juice is produced and gastric juice contains HCl. HCl removes this extra polypeptide and it converts pepsinogen into active pepsin. And then that active pepsin, it starts to digest the protein if protein is present in the diet. If not, then it is not activated. Right? Keep it in mind. Now, this was an exception. If there is no apoenzyme, there is no cofactor needed in this case. However, for its activation, the extra polypeptide has to be removed. Now, the types of cofactors, we have two main types of cofactors. How many types? Two types. We have two types of cofactors, or we can say three types of cofactors. Right? To be very specific, I can say that there are three types. Cofactors can be inorganic or organic in nature. They can be inorganic or they can be organic in nature. If they are inorganic, for example, zinc, right? For example, zinc, iron, chloride, these are all inorganic, right? They are inorganic in nature. These are known as activators. they attach with the enzyme, activate the enzyme, and then fall off. If they are organic in nature, then we call them the coenzymes. Then they are known as the coenzymes. They are, keep it in mind that these are loosely attached to the enzyme, and then they are loosely attached to the apoenzyme. They carry on the reaction, they bring about the shape of the active site, which most of the time in this case it is not fitting with the substrate. So these cofactors, they give it the suitable shape, the binding site, and then once the substrate is converted into the product, then they fall off and they go, and then they go and find another enzyme, that the another substrate and enzyme that they have to activate. Now, in a case where it is very tightly attached, in that case, it is known as the prostatic group. It is known as the prostatic group, right? Let me go back to the presentation now, and we can get the presentation and we can see what we have in the presentation. This was the second part of the lecture of enzymes, and I will be uploading more lectures. I will try to upload lectures regularly for you people, because I have been receiving messages from some students, but because of some extra tasks, I could not upload videos regularly, and there has been a break of about 2-3 weeks. And sorry for that break. Okay. Cofactors, activators, coenzymes, and prostatic groups are cofactors. What are cofactors? They are a non-protein substance which is essential for some enzymes to function efficiently. Activators assist in forming the enzyme substrate complex by molding either the enzyme or the substrate into a more suitable shape. For example, salivary amylase needs the presence of chloride ions before it can catalyze starch into maltose. Then we come to coenzymes. Coenzymes are also non-protein organic substances. They are essential for the functioning of enzymes. They are loosely bound to the enzymes. For example, nicotinamide dinucleotide. Nicotinamide adenine dinucleotide is a cofactor which is acting in the electron transport chain. It is a member of the vitamin B complex, and it acts as a hydrogen acceptor, to dehydrogenase the electron transport system of respiratory enzymes. And how is it? We have some other coenzymes also, like ATP, like FAD. This is the electron transport chain. From glycolysis and Krebs cycle, this is what is happening in the intermembrane space of the mitochondrion. These are the cofactors. These are the coenzymes: NAD, flavin adenine dinucleotide, nicotinamide adenine dinucleotide, adenosine triphosphate. These are all going to act as coenzymes. They will be loosely attached and then they will leave the product. Then we come to prostatic group. Prostatic group are non-protein organic molecules that bound very tightly, covalently to the enzyme molecule. You can get a question to write in your paper, differentiate between the prostatic groups and the coenzymes. Coenzymes are loosely attached, prostatic groups are very strongly bound together. And a very good example of this prostatic group is the heme group of hemoglobin. This heme group of hemoglobin, it is very tightly attached to the hemoglobin polypeptide chains. To the hemoglobin chains, it is very tightly attached to them. It does not leave the hemoglobin in any situation. It is covalently bonded to the hemoglobin. A very strong group, the heme group. Now look at this. This is the iron, which is attaching in the head of the hemoglobin. This is the head of the hemoglobin. And in chloroplast, we have magnesium in this position. That is also very strongly and permanently bound. Magnesium in chlorophyll, and iron in hemoglobin. These are examples of prostatic groups. Now, students, this was all for the lecture today. I hope you people keep on coming to the channel. Recommend to your students, to your friends to join the channel for clear and very easy concepts of biology. See you people next time. Have a good day.